Mm. Hello, everyone. Welcome to this week's Global Immunotalk. And it's really my great pleasure to introduce today's speaker, actually a good friend of mine, Dr. Lilin Ye, from Institute of Immunology, Third Military Medical University in China. Before, as Euro, that we always ask our speaker, a sweet question of their choice. But before that, let me briefly introduce Lilin. He did his PhD in immunology in University of Maryland. And he moved to Emory University to join Rafi Ahmed's group as a postdoctoral fellow. Then he returned to China in 2013, and ever since then, he's been focusing on studying CD8 T cells, so the differentiation, exhaustion, and its role in various disease, particularly most recently in cancer. And he has published extensively. I think one of the most notable contribution that he was among the first to show to identify TPEX cells, and also to show the tumor antigen-specific memory CD8s in the draining lymph node that can sustain an anti-tumor effect of the checkpoint therapy with the anti-PD-1 or PD-L1. So I think that's really a major contribution he's made. Our understanding of the trajectory of T cell exhaustion and provide some really important insight into the development of novel immunotherapeutic strategies that against viral infection and cancer. I believe he will discuss more in today's talk. But before I let him to tell us about T cell differentiation and exhaustion in cancer immunotherapy, I'd like to let Lilin answer a question that I think he has been pondering sometime. What that would be that if you could mentor your younger self, what would you say? I think that's also something that any of the young scientists in the audience would be interested in learning from you. Please, Lilin.
Okay, thank you for your great question. If I could mentor my younger self, I would offer 3 key pieces of advice. First, be patient and stay focused on your goals. Meaningful breakthroughs take time. Second, prioritize strong collaborations. Good science, generally comes from teamwork. And finally, worries, consider the impact of your work. Continually ask yourself whether your projects will deepen our understanding of immunology or contribute to innovative strategies for disease control. That's what I have for, if I could have a chance to mentor my younger self. Thank you.
I thought you have done tremendously well in that 3 regards.
Thank you.
Your younger self already knew those. Anyway, okay. Without further ado, I will let you to share the screen and tell us about the CD8 T cell differentiation and the cancer immunotherapy. Please, Lilin.
Okay. Thanks, Hai, for your generous introduction, and also thanks to the organizing committee for the kind invitation. It's really my great honor to present my work in this fantastic seminar series. Today, I'm going to do with the topic regarding CD8 T cell differentiation and cancer immunotherapy. Here, just showing you the early and brief history of CD8 T cells. Immediately after the discovery of T cells by Jackson Miller, Browner, and others, have realized that a subpopulation of T cells are cytotoxic, capable of killing their targets in 1970s. 5 years later, using monoclonal antibodies, Cantor and others found that CD8 T cells expressing found that T cells expressing CD8 molecule are cytotoxic. Later, immunologists have found that CD8 T cells recognize MHC class 1 restricted peptides using their T cell receptors. In 1996, peptide technology has been developed, allowing us to directly analyzing antigen-specific CD8 T cells. With these landmark studies, we can now distinguish CD8 T cells from other types of immune cells administered by two aspects. First, CD8 T cells can specifically recognize and serially kill their target cells, including virus-infected cells and malignant cells. Second, after antigen clearance, it generally leaves behind lasting multiple layers of memory CD8 T cells, including central memory, effector memory, and tissue-resident memory. These memory cells can provide long-term protection against the viral reinfection and the malignant recurrences. Therefore, antigen-specific CD8 T cells play a central role in antiviral and anti-tumor immunity. Current understanding of CD8 T cell differentiation indicates that during acute infections or vaccinations, activated CD8 T cells initially differentiate into effector cells. After the antigen clearance, most of the effector cells died, but a small fraction of those effector cells, also known as memory precursors, further differentiated into functional memory CD8 T cells. Upon re-exposure to antigens, these memory cells can quickly convert to effector T cells and rapidly clear reinfection. In sharp contrast, during chronic viral infection and cancer, where antigens persistent activity the CD8 T cells typically differentiate into terminally exhausted CD8 T cells. The concept of CD8 T cell exhaustion was first proposed by Rafael Ahmed's group in the mouse model of chronic viral infection, in which they found that virus-specific CD8 T cells were still there, but with much compromised effector function as well as proliferative capacity. Such features were then defined as T cell exhaustion. Later, T cell exhaustion has also been observed in human patients of chronic HIV, HBV, and HCV infections. Afterwards, T cell exhaustion has also been found in different types of human cancers. Buffett's group further demonstrated that compared to functional memory T cells, exhausted CD8 T cells express higher levels of PD-1 at both mRNA and protein levels. Critically, blocking the PD-1, PD-L1 pathway using monoclonal antibodies can restore the functionality of exhausted CD8 T cells and reduced viral titers in infected tissues, marking a great advancement to PD-1 based immunotherapy. When I established my own lab in China after leaving Rafael's lab, I was intrigued by a pretty much open question. That is, which subset of exhausted CD8 T cells can virtually respond to PD-1, PD-L1 immune checkpoint blockade, shortened as SAB. To cut the long story short, in 2016, including my own group, several groups identified a subset of exhausted CD8 T cells expressing surface mark CXCR5 and transcription factor TCF1 during chronic viral infection. This subset was then given a lot of different limbs, such as memory-like, stem-like, follicular-like, and progenitor exhausted T cells, shortened as TPEX. Whatever the limb is, we are literally talking about the same T cell subset. During my talk, I will refer to this subset as TPEX. Using CXCR5 RS-GFP report mice, we transferred GFP positive or GFP negative virus specific CD8 T cells into chronic infection matched recipients and found that GFP positive cells gradually differentiate into GFP negative cells, where GFP negative cells can never become GFP positive cells, indicating that CXCR5 positive TPEX cells function to replenish terminally exhausted CXCR5 negative cells. Later on, TPEX cells have also been found in other chronic viral infections. Moreover, TPEX cells have also been identified in different preclinical cancer models and human cancers. Beyond chronic viral infection and cancers, TPEX cells have also been found in autoimmune disease and allogeneic organ transplantation. Getting back to chronic viral infection, when adoptively transferred into chronic infected mice, only CXCR5 positive, but not CXCR5 negative virus-specific CD8 T cells can synergize with anti-PD-L1 ICB to maximally bring down viral titers in both spleen and lung tissues, indicating that virus-specific TPEX cells major ICB responders. This point also holds true in tumors. In 2019, one immunity paper and one Nature Immunology paper reported that TCF1 positive TPEX cells primarily respond to PD-1 ICB in different preclinical tumor models. These TPEX cells have also been found to positively correlate to the clinical outcomes of PD-1 ICB treatment in cancer patients. To conclude this part, basically, TPEX cells possess three distinct features. First, TPEX cells, stem-like, capable of self-renewing, thereby continuously replenishing terminally exhausted T cells. Second, this subset can be induced in disease settings with persistent antigens, such as chronic viral infection, cancer, autoimmune disease, and allogeneic organ transplantation. Finally, TPEX cells are primary responders to PD-1, PD-L1 ICB due to their stem-like properties. The discovery of TPEX has led to the acknowledgement that PD-1 ICB may act primarily on pre-existing TPEX cells within the tumor microenvironment. However, this notion is being challenged by two key observations. First, accumulating evidence underscores the importance of systemic CD8 T cell responses in PD-1 immunotherapy. And the second, systemic CD8 T cells are primed in tumor-draining lymph nodes or TDLNs. And several studies indicated that TDRNs very important for effective PD-1 therapy. However, the differentiation state of tumor-specific CD8 T cells in TDRNs remain poorly understood. To address this gap, we surprisingly discovered that a significant proportion of tumor-specific CD8 T cells in TDRNs are bona fide memory T cells. These cells expressing the memory marker TCF1, but not expressing the exhaustion marker TPEX, and then lacking exhaustion-associated genetic scars. These memory cells also display superior anti-tumor efficacy of the adoptive transfer when compared to traditional TIL therapy. Additionally, PD-1 ICB can expand these cells and drive their further differentiation into TPEX cells within TDLNs, which subsequently migrate to tumors and replenish this subset. I will refer to this population as TTSM cells, standing for tumor-specific memory CD8 T cells in TDLNs. Just 1 second. Okay. From these findings, we may propose that PD-1, PD-L1, ICB targets multiple layers of tumor-specific CD8 T cells, spatially and temporally. Essentially, PD-1 ICB can expand both TTSM cells and TPEX cells within TDLNs, driving their differentiation, expansion, and mobilizing their progeny into tumor tissues, thus continue to mediate anti-tumor effects. Another important point I would like to make here is that PD-1, PD-L1 monotherapy seemingly failed to change the differentiation trajectory from TDR and TTSM all the way down to the terminally exhausted T cells in TME, hindering the de novo induction of tumor-specific memory T cells. This limitation may partially explain the frequent tumor recurrence observed for the PD-1 immunotherapy. Looking ahead, we propose that combining PD-1, PD-L1, ICB with other modalities could divert TTSM cells from this exhaustion trajectory. Similar to response in acute infections, such a combination should drive TTSM cells to first differentiate into functional effector cells that can effectively eliminate tumor cells. Subsequently, a fraction of these effector cells should differentiate into secondary tumor-specific memory T cells, potentially offering long-lasting anti-tumor immunity and reducing tumor recurrence. But what factors could facilitate such process? We were drawn to several studies published in 2020, including those by Dr. Feng Shao at NIPS China and Dr. Judy Lieberman at Harvard Medical School, which suggested that tumor cell pyroptosis might enhance PD-1 ICB mediated anti-tumor effects. In brief, pyroptosis involves the cleavage of various GSDMB proteins by corresponding enzymes releasing their N-terminal domains, which forms pores in the cell membrane and lead to pyroptotic cell death. Among those GSDMB enzymes, GSDMB A and GSDMB B are particularly noteworthy because PD-1-ICB has been shown to enhance the expression of both enzymes in tumor-reactive CD8 T cells. This enhancement presumably increases their capacity to induce tumor cell pyroptosis. And more importantly, tumor-reactive CD8 T cells induce tumor cell pyroptosis through specific TCR, tumor antigen recognition, sparing normal cells, representing an ideal approach to future clinical applications. As I just mentioned, GSDMB or GSDMB had been implicated in increasing PD-1-ICB's effectiveness in controlling tumor growth. But here, we sought to investigate whether GSDMB or GSDMB pyroptosis could synergize with PD-1-ICB to induce tumor-specific memory T cells for long-term tumor control. To this end, we first engineered MC38 colorectal carcinoma cell lines to express either wild type human GSDMB or GSDMB, as well as mutant GSDMB, which is resistant to GSDMA cleavage. We inoculated mice with different GSDMB expressing MC38 tumor cells and treated tumor-bearing mice with anti-PD-1 monoclonal antibodies. Notably, where ICB intended the survival in MC38 GSDMB-bearing mice, such effect was kind of transient. In striking contrast, ICB resulted in almost complete tumor remission and long-term survival of 80% MC38 GSDMB-bearing mice, surviving beyond 200 days after tumor cell inoculation. Consistently, we observed a higher frequency of tumor-specific CD8 T cells in the MC38 GSDMB tumor tissues compared to those expressing mutant GSDMB or GSDMB following ICB treatment. Furthermore, we noted an increase in interferon gamma and TNF alpha dual producing effect-like CD8 T cells in MC38 GSDMB tumors post-ICB treatment, which is in line with enhanced tumor control in these mice. Additionally, we documented the progressive accumulation of toxin-negative CDC2L positive and CD127 positive tumor-specific memory CD T cells in the blood of MC38 GSDMB-bearing mice cured by ICB treatment. The combination of GSDMB pyroptosis and ICB treatment also induce long-term tumor control in other preclinical tumor models, including melanoma, small lung cancer, and triple-negative breast cancer, as compared to the combination between mutant GSDMB or GSDMB pyroptosis and ICB. Consistently, we can also observe the progressive accumulation of toxin-negative CDC2L and CD127 double positive tumor-specific memory-like CD T cells in the blood of GSDMB expressing tumor-bearing mice cured by ICB treatment. To further characterize the molecular features of tumor-specific memory-like CD T cells in B16 GSDMB-bearing mice post-ICB treatment, we performed single-cell RNA sequencing on tumor-specific P14 cells, recognizing the LCMV glycoprotein, which is incorporated into B16 GSDMB or B16 GSDME cell lines. Consistent with flow cytometry data, a unique memory-like subset emerged exclusively in B16 GSDMB tumor tissues, but not in those expressing either mutant GSDMB or GSDME after the ICB treatment. PCA analysis further indicated that this memory-like subset differed markedly from conventional memory subsets, including TCM, TEM, and TRM T cells. GSDMB analysis further showed that this unique memory-like CD8 cell subset displayed enhanced circulation-associated gene signatures, but reduced tissue-resident associated gene signatures compared to other naive, effector, or memory subsets. Flow cytometry data also confirmed these findings. The unique memory like CD8 T cell subset express central memory cell markers such as CDC2L, CCR7, and transcription factor TCF1, but not expressing TRM-associated markers such as CDC9, CD103, transcription factors lymph1, and RUNX3. And additionally, this subset showed enhanced expression of the circulation marker S1, PR1 compared to other memory subsets. Both CDC2L and S1 PR1 is regulated by the transcription factor KF2, which was also enhanced in this unique subset. Therefore, we can define this memory-like subset by target negative, KF high, CDC2L positive, and S1 PR1 positive cells. To assess the circulating characteristics of CDC2L and S1 PR1 double positive memory-like T cell subset, we first induced this subset through PD-1 ICB treatment in B16 GPU, GSDMB berry mice. Then we resected the tumor masses and transplanted them into resected mice. 10 days post-transplantation, we observed a wide tissue distribution of S1 PR1 positive, CDC2L double positive memory-like CD8 T cells across both lymphoid and non-lymphoid tissues, indicating their pan-tissue circulating feature. We also conducted pyroptosis experiments to validate the pan-tissue circulating capacity of this memory-like CD8 T cell subset. After inducing this subset, we conducted tumor masses transplantation and subsequently performed pyroptosis surgery. Consistently, donor-derived memory-like CD8 T cells were distributed across lymphoid and non-lymphoid tissues over the resected mice post-surgery. Based on these findings, we will refer to this unique memory population as pan-tissue circulating memory T cells, or PTC-TM cells. We also compared the migratory properties of central memory cells with PTC-TM cells. After the adoptive transfer into naive B6 mice, we found that TCM cells were largely confined to lymphoid, to blood and lymphoid tissues, where PTC-TM cells appeared in both lymphoid and the lung lymphoid tissues. To further explore the role of PTC-TM cells in long-term tumor control, we constructed KRF2 DTR mice and crossed them with OTR mice. We then transferred the DTR expressing or wild type OT1 cells into CD8 knockout mice, followed by inoculation with B16 over GSDMB tumor cells and ICD treatment. 45 days post inoculation, tumor-free mice were treated with DT toxin to deplete PTC-TM cells. Following this depletion, we can observe that tumor recurrence then occurred, indicating the essential role of PTC-TM cells in mediating long-term anti-tumor immunity. Furthermore, given the widespread tissue distribution of PTC-TM cells, we hypothesized that these cells could prevent digital tumor metastasis. To test this, we transferred PTC-TM cells, TTSM cells, or TRM cells into CD8 knockout mice and established models for liver and lymph node metastasis. The results pretty clean. You can observe that PTC-TM cells can prevent tumor metastasis in liver, and the lymph node, indicating that PTC-TM cells possess a superior capacity to prevent tumor metastasis compared to either TTSM cells or TRM cells. Next, we investigated the mechanisms underlying the induction of PTC-TM cells. We focused on conventional type 1 dendritic cells, CDC1 cells, which are known to be critical in determining CD8 T cell differentiation phase. We harvested CDC1 cells from GSDMB or GSDME present tumors after ICD treatment and performed single cell RNA sequences. UMAP analysis revealed 3 tolerogenic and the 2 sedimentary subsets. Notably, sedimentary CDC1 subsets were highly enriched in GSDMB present tumor tissues following ICD treatment. And the flow cytometry further confirmed that PD-L1 low, CD14 low sedimentary CDC1 subsets were markedly induced in GSDMB present tumors compared to GSDME counterparts after the ICD therapy. We then assessed whether PD-L1 and CD14 double low CDC1 cells facilitate the differentiation of PTC-TM cells in vitro compared to their high counterparts. Low CDC1 cells drive the more vigorous expansion of tumor-specific CD T cells. More importantly, they significantly promoted PTC-TM cell differentiation, shown as CDC2L and S1PR1 positive cells. We then hypothesized that a double transfer of double low CDC1 cells from GSDMB present tumor tissues after ICD treatment could rescue PTC-TM cell generation and thereby support long-term tumor remission in mice bearing B16 GSDME present tumor with ICD treatment. Indeed, this transfer significantly increase the number of PTC-TM cells and prolonged the survival in GSDME present tumor bearing mice. Finally, we considered how to translate these findings into the clinic, especially given that most of the human cancers actually downregulate GSDMB expression. To this end, we leveraged the tropism of avian Newcastle disease virus, NDV, for tumor cells, which had been widely used as oncolytic viruses to express GSDMB or GSDME. We confirmed the NDV expression and the GSDM protein expression in tumor cells by immunofluorescence and Western blot respectively. To evaluate the anti-tumor efficacy of the combined therapy with NDV, GSDM, and the PD-1 SAB, we established an orthotopic 41 breast cancer model and conducted different combination therapies. Remarkably, the NDV, GSDM-B, and the PD-1 SAB combination most effectively inhibited tumor growth, leading to about 80% of mice achieving completed tumor remission and surviving beyond 200 days post the tumor cell inoculation. And post-therapy assessment also revealed the presence of tumor-specific CDC2L and S1PR1 double positive PTC-TM cells across a wide range of lymphoid and non-lymphoid tissues on day 200 post-tumor cell inoculation. Consistently, the combination of NDV, GSDM-B, and SAB effectively induced CD14 and PD-L1 below CD7 cells compared to the combination of NDV mutant GSDM-B and SAB. The increased CD14 and PD-L1 below CD7 cells may facilitate the more efficient generation of PTC-TM cells, which potentially provide long-term anti-tumor immunity against tumor recurrences. To confirm this point, the 41 tumor-free mice receiving combo therapy with NDV, GSDM-B, and SAB were again subjected to either in situ tumor rechallenge or induction of liver and lymph node metastasis. It turned out that these tumor-free mice exhibited potent anti-tumor immunity against in situ recurrence and digital metastasis. Next, we sought to investigate whether the combo therapy of NDV, GSDM-B, and SAB could induce human PTC-TM cells. To this end, we first used NSG mice with a humanized immune system, which were then inoculated with a human melanoma cell line expressing cross with a G12V mutation and subsequently treated with the combination therapy. Consistently, the NDV, GSDM-B, and SAB combo therapy effectively induce human cross G12V specific PTC-TM cells and resulted in complete tumor elimination. And lastly, we investigated whether this combination strategy could also induce tumor-reactive PTC-TM cells in cancer patients. We initiated a clinical trial to assess the safety and the efficacy of this approach, recruiting patients with hepatocellular carcinoma and liver metastasis from colorectal cancer and breast cancer. Following 3 cycles of treatment, we observed the emergence of tumor-reactive PTC-TM cells in both the blood and the tumor tissues of patients. Additionally, we have evaluated the clinical outcomes in several patients receiving this combination therapy, and the early results look very promising. To conclude my talk, we propose that targeting GSDMB type of pyroptosis may serve as one of the critical factors enhancing PD-1 ICB's ability to induce tumor-specific pan-tissue circulating memory CD8 T cells, thereby facilitating long-term tumor control. Finally, I would like to express my gratitude to all my lab members who have contributed to this project. Also, special thanks to my close collaborators, including the host, Professor Chi, for their very, very important support. Finally, I wanted to extend my appreciation to the Tompin Laboratory for their great support to the clinical research. Thank you for your attention.
Thank you, Lilin, for that great and inspiring presentation. I think as a tradition, the Global IMMUNOTALK doesn't have a question and answer session. So the audience are welcome to post their question on the YouTube link, and I'm sure Lilin will be happy to answer a question in due time.
Yes, absolutely.
That will conclude today. Yeah, okay. That will conclude today's seminar. Thank you for listening, and thank Lilin for the great presentation.
Okay, thank you. Hi, have a great night.